In this video, I'd like to discuss enzymes. Enzymes facilitate biochemical reactions, and this is actually one of the major functions of proteins within our bodies is to act as enzymes. So what are enzymes exactly? So enzymes are catalysts. Catalysts are a substance that increases the rate of chemical reaction without being used up or changed by the reaction. So without enzymes, a lot of the chemical reactions that would occur within our cells would just simply occur so slowly that we'd be dead. It's just impossible to support life otherwise. So there are different types of enzymes, and we'll be discussing throughout this course many different enzymes. But let's look at just some simple examples here. One is a type of enzyme that breaks apart molecules. And the example I'll use is an enzyme found in saliva that breaks down starch. Recall that starch is a complex carbohydrate. It's a polymer of glucose, so it's a very highly branched, large polymer of glucose. You take in starch when you eat, say, a potato chip, French fry, corn. There's a lot of starch in lots of vegetables. And the starch represents a good food. It has a lot of energy locked up within those starch granules. So how do we address that? Well, the process of digestion actually starts in the mouth. You're mechanically chewing the food. That's part of digestion. But you're chemically also beginning the process of digesting starches, but with saliva. So you have salivary glands. They deliver saliva to the mouth. This saliva contains salivary amylase. That's an enzyme that breaks down starch. So we start to break down these large starch molecules into smaller glucose molecules, and that actually helps in the digestive process. So let's go on. Another type of enzyme is an enzyme that joins molecules together. So an enzyme may take two reactants and join them covalently. So let's take a look at how this occurs. You have reactants that bind temporarily within a site on the enzyme called the active site. So let's look at this picture here. We have an enzyme, we have two reactants, a yellow one and a green one. Now they'll actually go and fit into this active site, which is sort of like a trough or an area on the side of the enzyme. Recall that I mentioned in a previous video, the enzymes aren't static. They're actually able to change their shape a bit. And so what they'll do is once those two reactants are in place within the active site will actually move. The whole protein will change its shape a bit, and it'll bring the two reactants together. And this will help the reaction go forward, in which we have perhaps a dehydration synthesis reaction. Water comes off. The molecules are now, the reactants are now covalently bonded together. So once this product is made, it then leaves the active site, and you'll notice that we essentially can start the whole process over again. The active site is now available for the two reactants, the yellow and the green reactants, to then take their places within the active site. The protein then changes its shape, bringing the two reactants together. Dehydration synthesis occurs, and so forth. And you can see this can go on many, many times. In fact, so the whole point here is that enzymes are not used up or altered by the reaction. It can repeat the process many, many times. And in fact, this reaction can go forward at a very high rate. It can literally, an enzyme cannot make a reaction that wouldn't occur, but it can make the rate of the reaction much different than what it would be otherwise. So for example, without this enzyme, perhaps these molecules would occasionally collide with each other and dehydration synthesis might occur, and then they would, a covalent bond might be formed, but the rate of reaction might be extremely slow in, say, a test tube or something like that. Now you add the enzyme in, and the rate of reaction might be 1,000,000 times faster. So it's just a tremendous increase in the rate of reaction. And you have to think about not a test tube, but our own cells. Within our cells, we have many enzymes that are helping to make all these sorts of reactions go forward. And without these enzymes, you would simply be dead. The chemical reactions would occur too slow for life to be supported. Okay, that's it for this video. Thanks for watching.